Okay, thank you very much. So I would like to present the basically genetic study on Dupuytrens that we've started in Germany and Switzerland. So yeah, again, unfortunately we have no conflict of interest. And no conflict with the technique.
Say next slide. Next slide.
Would you switch the slide, please?
Next slide.
You're in the beginning.
Slide again.
Very good. Okay, so I would start briefly introducing the German study on Dupuytrens, then present our results on the epidemiology, and give an introduction, also a brief introduction to the concept of the genetic study, as you have in part already heard from other speakers before. So we're basically geneticists, and so the approach to Dupuytrens in our case is shown here. The Cologne University has a cluster of excellence on age-related diseases, and so this was our basic approach to Dupuytrens, working on age-related changes in cell biology and into the genetic basis for these changes. So we have set up a mostly German study group on Dupuytrens, and the different clinical centers involved are shown here on the map, including a number of universities in Germany and also partners from Zurich in Switzerland. And this is the state of the patient recruitment right now. We have collected into our database 560 patients, mostly from German and Swiss origin. We have documented a number of clinical findings, and I will present some results of this. We have used a clinical questionnaire, and I must say all these data are not retrospective data, but they were just collected for these actual patients. And we've also collected blood and tissue samples, and the current state of the project is that we have blood samples of about 500 cases. We have cultured cells of 280 cases and tissue samples of 230 samples. And we have, of course, looked at the age at onset, and this is actually the graph of the overview of the first surgery, as of course it is difficult to obtain the real age at onset. And we have a distribution of male and female affected of 8% males and 19% females. And we have a mean age at first surgery of 51 years of age, 58 years of age in males and 60 years of age in females. So I think the distribution, the difference between male and female affected is not only because of the earlier age at onset, as we have a clear predominance of male affected. The situation is relatively the same in familial cases. We again have the distribution of 1 to 4 between females and male patients. Only the age at first surgery is a bit earlier in the familial cases, around 55 years of age, both in male and female patients. In all, we have about 40% familial cases, with, of course, the limitations in obtaining this number. We have documented ectopic disease. We have 12% of the patients with ectopic manifestation, and in detail there are 6% of the patients with knuckle pads and 5% also with manifestation on the feet. We have also looked at a number of different associations with other diseases. Some of these are shown here. We have 10% of the cases with an association with diabetes mellitus, with a prevalence of 5.8% in Germany. So there seems to be an association with diabetes, as others have found as well. We have no association for instance, with rheumatoid arthritis, 3% of the cases with a prevalence of 4% in Germany, and we have 1% of the cases with an association with epilepsy, which is also very much the same as in the general population. We have furthermore looked at potential risk factors for Dupuytren's, and for instance, we have looked at smokers or former smokers, and we find 40% in male patients and 20% in female patients are or were smokers, which is even less than in the general population. These are data according to the German Federal Ministry of Health. And also, rather surprisingly, we have very much the same rate of alcohol consumers according to their own documentation, of course, very much the same rate as in the general population in Germany. We've also looked at other risk factors such as trauma and manual work, but these numbers are still too small to get a final result on concerning the epidemiology of these risk factors. We've then started also to look at the tissue samples and cell samples, and this is just one very preliminary result concerning the activity of the cells that we have obtained from these cases. We have cultured myofibroblasts, and we have seeded myofibroblasts into a collagen matrix, and then cultivated the system over a couple of time, over 2 months actually, but these are the results for 10 days, and you can clearly see the contractibility of the myofibroblasts in the collagen matrix, which is much higher in patient cells than in control cells shown here. This in the middle is just the negative control. This is the collagen matrix without any seeded cells. And so putting this into a figure, you can here, the blue bars represent the patient cells, and you can clearly see that the contraction is much higher in patient cells than compared to the control cells in yellow. However, the main purpose of the study is the analysis of the genetic susceptibility. And so let me just briefly introduce the concept for this, as others have done before, and Peter Dollmanns will probably do this afterwards again. So we are looking at a combination of environmental factors and risk alleles, and only the combination of these different partners lead to the manifestation of a multifactorial disease such as Dupuytren's. And the purpose of the genetic study, of course, is the identification of the risk alleles, of some of the risk alleles that contribute to the manifestation of the multifactorial disorder. And there are a number of different approaches to this way of gene identification, and we are following 2 ways. The first one is a whole genome gene expression profiling, and therefore we have basically obtained RNA from tissue samples. We will also compare the results to RNA from cultured cells with all the problems that were documented about cultured cells and their changes in expression. So the main focus will be on RNA from the tissue samples. And this will also give the opportunity to compare different disease stages, different culture conditions concerning the cells, and also, of course, age-dependent changes. The expression analysis is done using Illumina expression chips microarrays, which represent almost 50,000 probes. And the second part is then the whole genome association study. And so I would again briefly introduce the different concepts for the analysis of whole genome analyses, whole genome scans, with the 2 major concepts, linkage analysis or association study. This is the linkage analysis, which is based on families where the phenotype is segregating. So we look at only a few generations within a family, which leads us to rather large haplotype segments shown here. And we need accordingly only relatively low marker density. In contrast, when looking for association, we have an overview of many generations, and we obtain relatively small segments, which of course gives a relatively precise localization of the gene. However, we need a very high marker density, which is possible these days, for instance, using SNPs, using single nucleotide polymorphisms. And this is what we are doing. We have started the analysis based on a case control design with 400 patients. Actually, we have done 500 patients right now. And we've also used the SNP array 6.0 from Affymetrix, which was mentioned before, which also represents both SNPs and copy number variations. And this lets us integratively analyze the data and also look for the potential geographic hotspots. The concept for this approach with relatively small numbers of patients is shown here in the review by Pearson and Manolio. This is a multi-stage study. This means that we start with relatively small numbers of cases, but many different SNPs, and then other stages for the analysis have to follow, using much larger number of cases, but using less SNPs. And so we can, using the microarrays, we can start with these smaller numbers of cases. And of course, this is also a matter of funding, that we have to start with small numbers of samples. And this study is now ongoing. Unfortunately, I cannot yet present any data. We are just about to complete this first whole genome scan, and I hope to obtain the first results very soon. So we have now 500 cases, and we have compared these to almost 1,700 control samples. So to give a perspective of these studies, of course, we hope to unravel, at least to contribute to the genetic basis of Dupuytrens using this approach, but also beyond Dupuytrens, we want to characterize processes which are involved in myofibroblast proliferation and differentiation, and in particular to characterize stress-induced and age-dependent molecular changes, and thus identify genes involved in stress response and aging of connective tissues. And last but not least, of course, we hope that this approach will contribute to identifying a curative approach to Dupuytrens. So let me finally acknowledge the people involved. Of course, this needs, as others has also demonstrated, this needs large collaborations. And this is my group in Cologne. Let me mention Kerstin Becker. She's my PhD student who did most of the experimental work. And this is where the research takes place. This is our laboratory building in Cologne. And this is Cologne. This is the Cathedral of Cologne in the background, which you can see everywhere in Cologne. And actually, this is the opera building in the front. Thank you very much.